Welcome everybody to the Wednesday afternoon lecture series. My name is Lorenzo Leggio and I'm a physician and scientist and PI at NIDA and NIAAA. It's really my distinct pleasure to introduce our WALS lecture today, Dr. Kim Janda from the Scripps Research Institute. Dr. Janda is the Ely Callaway Jr. Professor of Chemistry. He's also the director of the Worm Institute for Research and Medicine at Scripps, and is a scholar at the Skaggs Institute of Chemical Biology at the Scripps Research Institute in California. Dr. Janda received his PhD in organic and medicinal chemistry from the University of Arizona. Then he moved to California to what at the time was called the Research Institute of Scripps Clinic. Now that's the Scripps Research Institute. So Dr. Janda was a postdoc fellow there, and there he stayed until now. He became a full professor in 1996. Along the way, Dr. Janda has funded 4 companies. He has been the recipient of many prestigious awards. I will just mention 2 of them as examples: the Alfred Bader Award and the Portuguese Lectureship Award, both from the American Chemical Society. He's also a fellow of the American Institute of Chemistry. He's a fellow of AAAS, and he also received a honorary degree from the University of Helsinki in Finland. Dr. Janda is author or co-author of almost 600 peer-reviewed articles, and broadly speaking, his research spans from medicinal chemistry to molecular biology, neuropharmacology, and immunology. His work includes the development of methods for the detection and the protection against the chemical and biological warfare agents, the design and evaluation of catalytic antibodies and enzyme inhibitors, and then the application of these discoveries and the application of immunopharmacotherapy toward the treatment of obesity, addiction, and cancer. The title of today's WALS lecture is A Vision Engaging Pharmacokinetic Strategies to Treat Substance Use Disorders and Overdose. Kim, welcome to the NIH, and thank you so much for being with us today. And the podium is all yours. We look forward to your lecture. Thank you.
Thanks, Lorenzo, for that introduction. I've been doing research now for almost 37 years. Funded out of NIH. I've done a lot of different areas, but I thought I'd do something very unusual today, try to focus on one area. As people know, I kind of go off on different things over my career. So I'm going to tell you about our work on developing basically vaccines for substance abuse disorders. So give some idea of what the lecture is going to be consisting of. I'm going to talk about these drug vaccines. I give some basic tenets and the mechanisms of how these things work, reasons for developing the vaccines, and then I'll talk to you about several of our vaccines, but I'm going to stick mostly in the opioid area. So heroin, fentanyl, alterated heroin, fentanyl type drug vaccines, and then some monoclonal antibody work. And then finally, I'm going to stay with the idea of using biologics for treating substance disorders. I'm going to talk about a nicotine degrading enzyme at the end. So, vaccines have been in the forefront for the last couple of years with COVID, and I think it's probably worthwhile we go back to the beginning here. So, vaccines have a long gestation period where they've started back in the 1000. Chinese employed these for variolation. Most people have heard of the milk cow with Edward Jenner, who's considered the father of vaccinology. With the young boy who was vaccinated with material from cows. Louis Pasteur is widely considered the first person to make a laboratory vaccine. Moving forward, I think it's kind of interesting if you look at 1926, guanine alum adjuvants. So probably the most widely used adjuvant still today is over 100 years old, alum. So some of the difficulties moving forward in coming to new adjuvants. Looking obviously at the 50s with the Salk vaccine, I kind of put this in the same version of what COVID is. One person probably no one's really heard of is this gentleman named Hillman who worked at Merck. This person was underrepresented. He's not alive anymore. He's probably saved more lives than anyone in the world with his development of, I think, about 7 to 9 different vaccines he's did over the years, but widely undercredited for this group. So even though we're seeing all the stuff that's been done with COVID, Hillman saved many more lives with some of the vaccines he's developed. So our drug abuse vaccines aren't much different than the normal vaccines, I like to say, but there are some differences in that we have the drugs aren't immunologic in themselves. So what we have to do is try to train the immune system to recognize drugs as being foreign. And so there's really 3 components to make a good vaccine, and that requires some synthetic chemistry in the hapten and linker design, carrier protein, which is the T cell, epitope, and then adjuvants. And so if you kind of fail in any one of these 3 aspects, you're not going to make a very good vaccine. Those points are critical. Some of the basics in terms of antibodies, where we see like antibodies made against a biologic or a virus, against a bacterial or a virus. Amount of antibody you have is very important. But with these drug vaccines, it's not just the amount, it's the quality. So the affinity to the drug and then specificity. So you have to remember, because the drugs are binding to different receptors and they have high affinity. And if you can't generate antibodies with high affinity, it's not going to work. So not just how much antibody you generate, but the quality of the antibody. So in generating these things, the hapten should try to mirror the chemical structure as much as possible and not be wasted on irrelevant structures. And so with these drug haptens, we don't really have epitopes. I like to call them chemical determinants that we're trying to use to generate an immune response. And so the haptic structure, depending on what you're trying to look at, has to be stable or not, depending if you're looking at multiple different displays. The linker is also been kind of pushed aside, but it's very important because how you display the drug to the immune system, and there's no doubt that the linker still shows some effects. The drug will bind the linker, so it's important to try to mask this in some way. And so, like I call it, the regiochemical placement is important. How flexible the linker is, that's not really something that has been determined. We've looked at things that are flexible and not flexible, and it can vary. How many copies you put on the hapten is important. So this really controls whether you're going to get a T cell dependent or T cell independent response, and so the efficiency. So I like to kind of mirror this with Goldilocks and the 3 beers: not too hot, not too cold, just right. So the best linker is between 4 and 8 atoms. That's what we've found over the years. So in terms of the immunological mechanisms, how these work is quite similar to a lot of other vaccines. So you're going to formulate the vaccine, inject it. It's going to be taken up by dendritic cells, eventually displayed on the MHC, which is going to help activate T helper cells, which then eventually is going to activate B cell receptors. And hopefully at this point, we're going to stimulate the B cells to make antibodies and potentially some memory from this. So the simplest way to explain this is we're generating antibodies. Typically, antibodies don't cross the blood-brain barrier, so they're going to basically block the drug from getting to the receptors in the brain. But you also have to realize, depending on the affinity, these in the drug's PK, the antibodies can withdraw the drug out of the brain. So it's like with Chatelet's principle. You can basically alter the pharmacokinetics of the drug in the brain by just pulling it out. So over the years, we've looked at many, many different drugs, but I like to put the main 4 that we've looked at. So nicotine, cocaine, methamphetamine, and then the opioids. So looking at the opioids, I like to basically group them into several areas, whether they're natural, semi-synthetic, or synthetic. You can see here in red, we're just going to talk about the semi-synthetic and heroin, and then the fentanyl analogs. So heroin's been around a long time. It was originally sold by Bayer as a relief for coughs, and as you can see there, it probably could relieve almost any cough. Heroin, in essence, is a prodrug, shown here. So The reason why it's acetylated is because the heroin itself crosses the blood-brain barrier much quicker than the actual drugs, that 6-acetyl-morphine or morphine. So in developing a vaccine for this, the question is, well, what do you want to develop antibodies against? Do you want to develop against heroin, which really doesn't have a lot of binding affinity to the mu opioid receptors, or 6-acetyl-morphine, which has much greater, or morphine itself? And then some of the other possible products from first pass metabolism. So that's critical, and I put the heroin's a moving target, so in terms of developing these vaccines. Okay, so I'd be remiss not to say that there aren't medications for opioid addictions. There's substitution therapy, and I think we're all aware of methadone and buprenorphine, and they have great advantages, and there's some disadvantages with these that I've listed. There's antagonist drugs like naltrexone, and that has advantages too. Probably the greatest disadvantage is most addicts don't like it because it gives them unpleasant symptoms from it. And then there's psychotherapy and cognitive behavioral therapy, but a lot of times this is insufficient because there's low compliance rates. So we think these vaccines can be of value in that it doesn't require daily compliance. We would hope that over time it could help normalize the individual. It should reduce the risk of overdose, avoid side effects, less costly to maintain. There shouldn't be any illicit diversion to other opioids. And I think one of the biggest things is scaled globally. And originally, when I was looking at developing these opioid vaccines a number of years ago, this wasn't for the US. This was before we had this pandemic. This is really for third world countries, where I saw an increase in opioid use with an increase in HIV. So people who were sharing needles back then. So I thought this would be something that could be viable for third world countries, not really the US. In designing these vaccines, we take a lot of time to look at how we want to develop and before we even go out and prepare them. And so there was a rich amount of literature on antibodies to heroin, but it was really from the standpoint of looking at ELISA, so binding, so testing for drugs if the person was taking the drug. And so looking at some of this, we came up with the best approach we thought would be putting the linker off that nitrogen there. We thought it presented heroin as the most natural conformation, and we thought it could give greater specificity and affinity for the drug. So we made two conjugates at this time. One was the heroin one you see with the red arrow, and the second one was morphine, because we wanted to test out, again, this idea of what you don't want to make two. Do you want to make the heroin or morphine? Now you have to also realize that this heroin conjugate we thought would degrade, so we could get antibodies to 6-acetyl-morphine and potentially morphine. So I won't bother people with the synthesis. This is one of our syntheses that we come up with. So these aren't things you just take the drug and voila, go immunize. Like the key step is really demethylation, and then you can see on the right, number 7 and 8 is the linker. So 7 would be the heroin-like vaccine, and 8 would be the morphine-like vaccine. So you see this STR, so that's a trityl group with the sulfur, and that was our initial approach, but we've gotten away from that. I'll show you a little bit later for several reasons. One, stability reasons. Also for conjugation, these tend to do what's called a retromichael reaction, and so they can come off. And so it's not really, I think, a great linker for moving forward if you want to do FDA type studies. So recapitulating design, we have a drug, we're going to do some synthetic chemistry on it. We're going to put a linker, have a functionality that allows this to a carrier protein. We're going to then formulate this with some type of adjuvant. Then we're going to vaccinate animals. We're going to look for titers, and then we're going to try to examine them in some behavioral models. So the first model we ever looked at was the hot plate test. And I should give credit. So when George Koob was back here at Scripps, and actually I worked with George for 30 some years, actually the drug abuse. So I should give George credit, because when I originally started working in this area in cocaine, I didn't know what to do in terms of animal models. I went to talk to George about this because I was supplying him with agonists or antagonists. I was failing miserably at the time. And so I told George I had this idea about vaccinating, and George just said to me, he goes, "Is it going to work?" And I said, it's going to work. So he said, okay. So George actually took me under his wing and ran most of the behavioral studies and taught us how to do this. So George deserves a lot of credit for this. So hot plate test, getting back to this, was we looked at heroin and oxycodone. So you can see on the left, we did the control, which is KLH, this carrier protein, heroin, the heroin vaccine, the morphine vaccine, and we did a combination of the two. So you see the heroin vaccine worked best, the morphine vaccine didn't work at all. So morphine was not really a critical player, we believe, in terms of developing these vaccines. And then you can see the other thing, which was quite surprising, where we looked at oxycodone, and basically these vaccines are extremely exquisite for heroin. Okay, so as I said, George helped us set up some more sophisticated animal models. And so one of the ones we had was an operant box where we had these rats in there where they basically learned to press the lever for drug. They could either be cue driven or not. And then, so basically this would take place over a period of about a month to get them acclimated to self-administration. Then we took them off the drug for about a month. We vaccinated them, then basically put them back in the box with the levers. So the key point, I'm going through this really quick, and George is probably, if he's watching, is probably squirming because this took months to run. Basically, you see three things. One, they escalate without the vaccine. Two, depending on cues or not, they either become static or they basically extinguish. So this was actually quite exciting at the time when we did this. Another key thing that we ran that George said we should do, and that is re-escalation. And so this so-called idea of motivation. So this is where they have basically unlimited access for a few hours of the drug. So pressing like 1, 1, 2, 2, 3, 3. So working harder, progressive ratio, taking the drug. And I should say we have seen in some of our vaccines, specifically nicotine, where they will surmount this and load up more drug. But we didn't see this with heroin. So this was, again, an exciting result. I'll just show you, this is a little video, for a sedation video, where we have about 10-fold more drug on board than the ones that are vaccinated with the ones that aren't vaccinated. So I think this gives a good picture of how well these vaccines can work. So obviously you can see the guy in the corner that's not vaccinated, got a couple mg per kg, where the other one's got about 20 mg per kg that's vaccinated. So when the vaccines are working well, they can work extremely well, as you can see here. So we weren't completely satisfied with this because of some of the work we've done with our cocaine vaccines. We thought we could try to improve upon this. And so one of the things we did a lot of work looking at different adjuvants. And one of the ones we came up with worked really well with CPG. This comes out of Pfizer. It's been in clinical trials, I think at least phase 2 clinical trials. And when using CPG, which is a TLR9 agonist, we got about a 10-fold bump in potency. Again, this is looking at hot plate or tail flick, tail immersion. We improved that a little bit more. Like I said, we removed that thiol and we put an acid out there, so COOH. And we then changed the carrier protein to tetanus toxoid. And so when you combine all these things together, we got about a 30-fold improvement in terms of shift to the right with regards to this vaccine. In moving forward, one of the things was, can we, how stable are these vaccines? And so, especially with the COVID ones now that you're seeing with these mRNA ones, RNA ones, is that they have to be kept cold. And so, especially if you're going to work in third world countries, you have to beat the cold chain. So I went off and decided to kind of play around with this. And I came up with this formulation where we use trehalose. And basically, if you have this formulation below, and you can have alum, the whole thing together, this thing is stable for months at room temperature. So that really mitigates some of the degradation. And we had some animals left over. We wanted to see how well it would look in terms of, protecting from overdose. And it does work to some extent. You can see not complete 100%, but that we did get some reasonable protection. So the key aspect is here, we can surmount the cold chain with this formulation. We've done some work with Matt Banks, Steve Negus out of VCU. Specifically, they had the ability to work with non-human primates. And I'm not going to go through this. This was Matt Banks' work, but we've done a number of studies with him. And so we were trying to compare this with naltrexone. Some days it worked better than others. What we really like to see is at least an 8 to 10-fold shift. Matt used to say in this terms if it's going to work in the clinic. Some days we see this, sometimes we wouldn't. One of the reasons why it works so well, we believe, is again binding affinity. So we did this by surface classes resonance spectroscopy. So you can see both monkey and mouse affinities remains pretty similar. And then on the right, you can see that, again, we have this exquisite specificity for this, that you don't see off targets like the oxycodone or methadone or fentanyl. But as I said, one of the issues we had with these heroin vaccines is that at the beginning, I was really excited about this, but as time waned on, we've been doing this, especially in non-human primates and some other rat models. I'm not sure how well they're going to hold up in the clinic, to be honest with you. And so I kind of went back to the drawing board to look at if we could improve upon this. And so one of the things I've looked at very recently is a thing called deuteration. So using deuterium on these vaccines, would it have an impact? So you can see the overall pharmacokinetics going from heroin to morphine. So what I wanted to do was the 3 and 6 position of those acetyls, I wanted to replace this with deuterium, which is known to slow down the pharmacokinetics of hydrolysis of this. So I should say deuteration is, if you look in the literature, there's almost nothing on deuteration in vaccines, whether it's a drug abuse vaccine, which is nothing, or even just subunit vaccines. So I think this could be a really fruitful area to work in. We'll see in the future, but we had to go about making this. So again, I won't go through the synthetic chemistry, but you can see 4 has the deuteriums, 3 deuteriums on there, where 7 is the original kind of vaccine. And so we did this kind of the cheap, so we didn't maximize the carrier proteins or anything like this because they're expensive. Just wanted to compare the two head to head, how this would work. It's just simple, 1 proton increase, what would have happened with the deuteriums, the CD3 versus the other one. And so we were actually quite surprised to see that not only were the titers increased, but we saw, well, there was differences between these M and F, male and female, but the vaccines could select between the deuterium versus the non-deuterium, the deuterium one. So we saw differences in titers. So that's very small perturbation. The immune system could readily survey and pick up. So I was kind of actually surprised to see this. And I wouldn't be showing it. So it did work. The deuterium vaccines, you can see DAC, they're in the orange, male and female. So the ratios, what we're looking at, the lower the ratio, this is between the blood and brain. You want to keep it in the blood versus the brain. The lower the number, the better. So you can see these deuterium vaccines are an improvement over the original vaccine. So maybe in the future, we'll look at this in a little more detail. But I think this could be a very important area. Okay, so that's kind of it with the heroin. I'm going to move on to fentanyl. So we started looking at fentanyl before it really became so-called in the news. This was a number of years ago. And I really decided to look at this because I wanted to really challenge the whole idea of vaccines, because fentanyl is so much more powerful in terms of binding the opioid receptors. So could we make a vaccine that really could alter its capabilities? So some of the fentanyl analogs, sufentanil, alfentanil, low fentanyl, carfentanil, these are all can be medically. Carfentanil is not. That's used basically by vets. So some of the ones there. These were made by Janssen in the 60s, and they're still used in anesthesia. So one of the key components I thought about in developing these fentanyl vaccines versus the heroin ones was I wanted to make a broadly neutralizing one. So heroin is specificity. The fentanyl ones, because there's so many designer ones out there, we wanted to see if we could make something that could broadly encompass a number of different fentanyl analogs. And really the reason why fentanyl comes up is it's based on profit margin with the drug dealers, so it's a way to compensate. If heroin gets impure, fentanyl creeps in. If heroin becomes more expensive, fentanyl comes in because it's fairly inexpensive to make and it's much more powerful. Another aspect of fentanyl is kind of underappreciated. It does bind the mu-opioid receptors much more tightly, but its rate of transfer from drug from plasma is pretty quick. So you can see here remifentanil versus morphine. It's got extremely quick transfer from drug from plasma. So that's another key aspect in terms of how reason why these work so potently and quickly. So in looking at the overall metabolism of fentanyl, like we did with heroin, fentanyl is actually pretty stable. The main route is this oxidative de-escalation, so norfentanyl. But so in terms of like, we didn't really have to worry so much about, you know, the hydrolysis aspects like we did with heroin. So really it's the question with fentanyl is focusing in where we want to place the linker so we could try to develop broadly neutralizing antibodies that would be cross-reactive with a number of different fentanyl drugs. So here again, it's the synthetic process we use. It's just a few steps. I won't go through it. And you can see why it's very easy to make fentanyl analogs where we can make this vaccine in just a couple steps. So our first generation fentanyl Aptin is shown here, and we looked at about 6 or 7 different fentanyl derivatives. So our timeline was eye or injection, and we're either testing for affinity, or we do blood-brain barrier distribution or antinociception for the A. So this was really gratifying to see this when we got this data. We saw massive shifts in the curve with these vaccines, much better than I thought we would see. 30-fold shifts. We see it found a number of different fentanyl analogs. The reason being is because we really keep the drug out of the brain, and basically control it into the blood. The other key aspect of this is these antibodies bind extremely tightly, you know, low nanomolar level to fentanyl. And again, we see nothing to MED is methadone or oxycodone that's basically doesn't see this. So extremely tight binding to fentanyl and a number of analogs. And then we just did just a quick one. We had some animals left about overdose, and here we saw complete protection with these vaccines. So these fentanyl vaccines are much better than the heroin vaccines, I should say that. This was actually a really interesting study. It was put out by NIDA a few years ago. I don't know these people, but came across it. I was really intrigued by this, by the addition of fentanyl, and they saw this, increase in hypoxia was potentiated by this mixture. It wasn't like a one-to-one thing. So I found this pretty interesting. So we decided to look at this from the standpoint of whether heroin being adulterated with fentanyl. So we had these 2 vaccines, the heroin vaccine and the fentanyl vaccine. So it was easy to do. We just mixed the 2 together. Now, the one piece here that's a little different is that the mixture of fentanyl and heroin, we use only 25 micrograms of each one, where the heroin and fentanyl, we use 50 micrograms. So they're not going to work quite as well as the individual ones. So as you might expect, the vaccine groups of fentanyl, heroin, they see either one well, they don't see the other drug well. The combo vaccine sees both drugs well. And this plays out again with the antinociception. The individual drugs themselves work better than the combo drug against each drug, but the combo drug works reasonably well. And this is also bore out in terms of the blood-brain barrier work that we did, distribution studies. Okay, so talk to you a little bit about some monoclonal work. And this kind of actually came about in discussions with Nora Volkow in terms of looking at some monoclonals for this. And she knew we were doing the vaccines. She was inquired, what about monoclonals? And I said at the time, the monoclonals will work, but it's really the cost of goods and producing them. But they have gone down quite a bit, and you can make monoclonals these days that have extremely long half-life. So I said, okay, we'll take this on and look at this and see if we could come up with a possible therapeutic. And this really goes, what we're looking for is looking at carfentanil. You can see the toxicity issues here. So this is shown in this slide here, where you can see fentanyl versus carfentanil in a penny. That carfentanil is extremely potent in terms of going forward. So with regards to antibody manifolds and looking at this, there's probably nowadays, there's like 100 different manifolds. So so the IgG, which I show on the bottom, that's what you typically generate from an immune response, a typical vaccine. And the Fab is basically a piece of that IgG, and the single chain is another piece. So the single chain contains what's known as the complementary determining regions where the binding takes place. It's 25K. The Fab has a little bit more. It's 50K KD, and then the IgG is 150 KD. So depending on what you want to look at, these manifolds, you can set up the half-lives or how it distributes in tissue or things like this. So you can play around with a number of different parameters in terms of these antibody manifolds, which is kind of makes it interesting. So initially when we did this, we looked at just simple hybridoma cell production. And this is based on work Kaler and Milstein, it's 73 or 75, when they won the Nobel Prize, where we're just going to vaccinate a mouse. We're going to take the spleen, fuse it with myeloma cell line, and just select for positive hybrids. And so in doing this process, you're only going to look between 30 to 50 monoclonals versus thousands and thousands, which you could generate. But this is a good tried true way, which I used for years in generating some of the catalytic antibody work we did. So we thought we'd go off on this first before we spend a lot of time and energy on some other methods. So in doing this, we were kind of actually pleased that we could make tight binding antibodies to fentanyl. And actually these antibodies bound, as you can see on the left, a number of different fentanyl analogs, not some of the ones like alfentanil or remifentanil, but some of the other designer drugs. So they bind quite well. So we went forward with this one called 684, which is right in the middle. We could have picked number one, but this expressed fairly well, so we could get large amounts of it. Dropping back a minute, one key aspect why these antibodies work so well is we look back again at Viacord type analysis. They all have very similar on rates. It's the off rate. So how slowly they come off the drug itself. That's the key point. So you can see they can vary quite widely. So we looked at some binding kinetics for this, and you can see these antibodies bind sub-nanomole level to both fentanyl and carfentanil, which is great. So that's in the ballpark that we're looking at. And we looked at this one antibody, like I said, 684, a little more detail. Its half-life is about 6 days. As you might imagine, it sequesters the drug in the blood, so it increases the drug half-life quite a bit. You can see in the left corner, so the free drug amounts in terms of how much the half-life is increased. So again, it's doing its job of sequestering the drug in the periphery. We looked at antinociception with regards to basically dose response. And so it is dose responsive. So we went from 30 to 120 mg per kg of antibody. So you might think 120 mg per kg is a lot, but actually this is still about 3 to 4-fold less molar basis of antibody to drug. So you don't necessarily need a stoichiometric amount of antibody to drug to work. We tried to do some rescue experiments using the antinociception model, and you can kind of see it, both the left is fentanyl and the right is carfentanil. We compared this with naloxone, and the overall goal here is because naloxone has a relatively short half-life to get renarcalization, we wanted to show that the antibody is not going to have that issue. And it's kind of blurs out a little bit here, but it's not quite what we'd like to see. So you'll see in some upcoming slides where we use cardiovascular depression models, and you can see this will come into view a lot better. So with the idea we could get good binding affinity to fentanyl and make these antibodies that appear to be fairly broadly neutralizing, we decided to try to go about making some humanized antibodies. And so you can humanize a mouse, but it's a lot easier just to take a transgenic animal where they produce these human idiotypes and then move forward with that. So we looked at this OmniRat because one of my colleagues, Dennis Burton, was looking at this with HIV, and he told me this would be a good model for this. So this is what we looked at. We had our conjugate. Basically what we're doing now is we're going to make what's called a B-cell library, shown here. I'll show you, we have these fluorescent probes, which allow us to do single cell sorting. We can pull out the clones of interest. We then characterize their binding. If it doesn't have the binding we want, we can try to make a library and improve upon this, or then look at antinociception, overdose. We were hoping to get to non-human primates, but we never got out that far. but that was the overall goal. So here are some of the probes that we made. So we made 2 probes. One was a carfentanil probe on the left. The other one's a fentanyl probe on the right. And this has biotin onto this. So what this allows us to do is attach it to streptavidin, which had basically fluorophores on this, and this allows you to do the sorting. So you can't just go off and sort. You have to make these probes to do this. So you can't read this on the slide, but you can see here we had antibodies to both fentanyl, carfentanil, some broad cross-reactivity, some not. So we got the whole gamut here. One of the things that was really gratifying is that we got extremely tight binding. So this 1A4, you can see this is sub-picomolar level to carfentanil. So this is like Guinness Book of World Record with regards to affinity to small molecules. So this is as good as you're going to get there. So this was actually really gratifying. And then they cross-react with fentanyl. We then did some pharmacokinetics on this. Again, depending on the dose of the antibody you use, we can get huge rightward shifts in the curve. Half-life is rather short with these antibodies. This is in a mouse, so it's okay. So this kind of got us to the point we were hopeful for, picomolar level binding to both carfentanil and fentanyl. One of the things that kind of threw us for a little bit of a loop was that in trying to move forward in terms of we're going to go in the clinic with these guys is stability. And you want to have an antibody in the blue on the bottom have a stability, a melting point temperature in the range between 70 and 80 degrees. And so we looked at this by DSC, differential scanning calorimetry, or DSF, which is fluorimetry. And so the key piece is to look at the TM1. These are in the mid 60 degree range. So this is not what you really want in terms of moving forward. And so we felt we had to try to improve upon this. So what we did was we turned to phage display, where basically you can link the phenotype to the genotype, so you can display antibodies at the surface of the phage and link this to the gene. This won the Nobel Prize in, I think, 17 or 18. My lab contribution, we basically held a patent on displaying on genes 7 and 9, where you hopefully see my arrow there. So we decided to employ that because I didn't want to be encompassed by patents looking at 3 or 7, so or 9. So that's what we looked at. We made some libraries off this. So basically what you do is you basically clone DNA fragments into this phage mid, plate it out, amplify, purify the phage, and then do some selection process. And then keep the binders, elute the non-binders, and then take the binders you want, go back through and go through this process several iterations. And at the end of the day, you want to last round of panning, you look to see, you express some of these clones and see how well they bind. Usually, this doesn't work very well, but in this case, it did work pretty well. So you can see here on the left where this 1A4 wild type, the single chain we used, which I showed in the earlier slides, had a melting point of about 31.5, where the mutant almost went up 20 some degrees. So this was really a really great result, actually. So moving forward, the question is, did we change its affinity greatly? So the wild type in green on the left versus the mutant, the yellow on the right, it did affect, if you can see the ratio somewhat, but it wasn't massive. So that was actually a good result. So we really increased its overall stability without really greatly stressing out its terms of binding affinity. Okay, so in looking at this, one of the things we were able to do was generate a crystal structure. We haven't published this, of this antibody. And you can see carfentanil bound there. But lo and behold, what we found was this antibody had a sugar on it, this lactosamine, you can see with that yellow arrow. And this is in the light chain. And this is a no-no in terms of moving forward with regards to manufacture. So it's actually, the good news is we got a crystal structure pointed as out. The bad news is that we got this sugar in there and we got to get it out of there. So here's another view of this here. So what we did was we went back into the phage display, and so that cysteine residue you see with that red scissors, we basically mutated that serine residue, either lysine, asparagine, or threonine, and we picked out the best clones which have favorable binding. So here we got another crystal structure which actually proves that we were able to remove the sugar, and in fact didn't really affect the binding any. And so here's actually the final crystal structure with all those mutations, which help its affinity, and you can see in the light magenta the carfentanil bound in. So this will be our guy in moving forward, this antibody here. So as I said, we're trying to look at this renarcalization type effect, especially with these extremely potent binding carfentanil. And the reason why you get the renarcalization is carfentanil is very fat soluble and it stays around. So when naloxone basically its half-life wanes away, carfentanil can kind of slowly slip back into the system and people can then become affected again because of its potency. So we spent a few years developing this model. This is our setup here. And it was really painful, but we're actually really good at doing this now. In fact, this is our model of choice. It takes time to do it, but the data you generate from this is much more, I'd say, robust with regards to versus the anti-nociception type work we did. So I think this gives us a really good feel of what's going on. So what we're looking at, there's a number of different parameters you can look at, but basically normal respiration going to respiratory induced depression, and we're trying to restore breathing. That's what we want to do. So here's a slide right here where we have naloxone on the left, where we inject carfentanil, we rescue. You can see with the bars that naloxone does increase it quite a bit in terms of the minute volume, but then it starts to drop off for a period of time. Where you can see on the right with the antibody, it basically comes in not quite as quickly at the rescue, but it basically is not dropping off. So you're not going to see the renarcalization with this. So this is a really fantastic result. I think this bodes well for trying to move an antibody forward in terms of looking at it for overdose type situation. So I just want to finish up with some work we've been doing on nicotine over the years, off and on. I think this is kind of interesting just to put perspective in terms of how antibodies work and the whole PK type phenomena that we're looking at. So I think most people know the nicotine problem in terms of trying to quit. A lot of people say it's more difficult quitting smoking versus heroin. Main pathways, there's a number of them, but dopamine is a key contributor to this. I want to point out that, NIDA and companies have invested, there's probably been 20 vaccines that have hit the clinical trials, and all of them have failed using the antibody vaccines. And to be blunt, they're not going to work. There's just, I don't care what you do, they just aren't going to work. And part of the reason is based on the PK with nicotine. You got to remember how these drugs work is that they're sequestering in the blood, and you got to eventually, it has to be processed. And what you're creating is basically a sea of drug. You're sequestering it. So what you really like to be able to do is degrade the drug. So if you could do this, sequester it and degrade it, then I think you'd have something very positive. So we're taking a look at this right now in the future is with regards to toxicity. So we're not really looking so much for this as cessation type in terms of nicotine treatment, although I have slides for this. But what I really like to look at this from is from a pediatric standpoint. So as we've seen over the years, vaping and the so-called liquid nicotine is unregulated. And I think you have to remember, I should have up there, nicotine IV is as toxic as fentanyl. It is extremely toxic. And so with regards to children, it's extremely deadly for them either vaping or drinking this stuff. It's not as problematic with adults. It's really based on your size and weight. But this is a real problem with children. And there is no therapeutic treatment for this at all. So this is where we're coming at when we look at this enzyme, is this aspect here. So in kind of perusing the literature over the years, I knew there was these, the bacteria that basically use nicotine as a sole source of carbon and hydrogen. They lived on nicotine. And the people are looking at this more remediation, like, in terms of water or soil, trying to use this in some format. But I was interested in, could we use these as a way to degrade nicotine for toxicity issues? And so one of my students had to renew his visa back in China, so I had him go back to one of these tobacco fields, and we got this and cloned it out. And basically, what we did was this first step, which is highlighted in blue, which we call that enzyme NICA2, which basically does an oxidative step, and then water opens up to pseudo-oxynicotine, which has no therapeutic effects. So that was the overall goal, was to try to take advantage of this enzyme. This enzyme is a really bizarre little enzyme in that it binds, you can see the red KM, really tightly. It binds nanomolar. So most enzymes are not in this class. So this is like the top 1% of enzyme binding. If you look at enzymes in general, like thousands of enzymes. But its KCAT's not very good. It's like what I would call a catalytic antibody. But it's KCAT or KM, its efficiency is about in the middle of what an enzyme can do. So it has some advantages, disadvantages. The one advantage is with the low KM, it's going to be saturated the whole time with regards to how much peak nicotine you see at any time. So that is an advantage with it. We got a crystal structure of this with Karen Allen at BU. It's a flavoprotein. And you can see there, it says we had 52 amino acids we couldn't find because it seemed to be flexible. And this actually came to our advantage because I'll show you in a minute how we truncated them out. So it's a flavo enzyme. It goes through an oxidative mechanism in terms of how it works. It generates peroxide is generated during the reaction sequence. And we did look at overall analogs. So you might imagine if it's the first committed step in terms of degrading nicotine, it better be really specific. And it's extremely exquisite specificity. It doesn't hit any of these closely related analogs. But as I said, the one unit was basically 52 amino acids was not visible, the crystal structure. So we decided to remove that and put in fuse an albumin binding domain. And what this allowed us to do is it greatly increased the half-life. So the original half-life of this enzyme was about a day, and we increased it to now to about a week. So that was a great improvement. And you can see here, we in this slide here, we dosed nicotine chronically for 5 days, gave the enzyme once a day. And you can see here, it basically obliterates nicotine. So both in the blood and brain. And we didn't do this, but Olivier George, who's now at UCSD, he looked at some more sophisticated models, both at withdrawal, which is A, pain threshold B, and irritability score C. And you can see the black was our enzyme, and it was favorable in all three aspects of this. So it worked pretty well. Okay, so that's all I've got for today. Just finishing up. Hopefully, I've been able to convey that we can get a robust immune response against these opioids. It's critical what you're looking at to look at what are the key breakdown products, especially with heroin. These vaccines won't block clinically useful opioids. We can develop those if pain is needed. I should say, I didn't bring this up, but these vaccines are what I call on par with COVID vaccines. So I used to get tremendous pushback from study sections and people at NIH saying, you're not going to get away with using these vaccines because it requires multiple boosts. I don't want to hear that anymore because of the COVID stuff where people are taking multiple boosts. This is just non-factor in terms of this. I think I presented a little bit of work with these deuterium vaccines. I think this could be quite exciting, even with proteins, deuterium, these things. I think these could have a drastic impact on how they work. We're working towards this heroin street vaccine. We can block synthetic opioids in a single vaccine. We're trying to work on ones where I have a hapten which contains both of them in one shot. I think you can see these antibodies, monoclonals, can be used for overdose treatment. And we could edit them in such a way that we could make them short lasting or long lasting. So they could maybe be used for cessation type therapy. And then just try at the end introduce the ideas that, well, simple binding is important, and using this PK pharmacokinetic idea, it works well. But if you could come up with something that not just only binds, but can degrade the drug, this could vastly improve a lot of the things that are out there. So let's leave it at that. So I think this last picture, this is actually looks like that today. It's a nice sunny day out here today. Group that did the work, I've already mentioned most of the collaborators. Very grateful to the work that I presented here was NIDA. It's work, SCAG's Institute, tobacco-related disease program out here in San Diego has supported some nicotine work. Like I said, I have worked in a number of other areas. Unfortunately, I didn't talk about a lot of stuff I've done with NIAID over the years, but I've also been grateful for their funding. So with that, I think we have a little bit of time. Lorenzo, I could answer a question or two.
Yeah, absolutely. Well, first of all, Kim, thank you so much. This was an outstanding lecture. And, you know, we are in the middle of an addiction pandemic. So really thinking outside the box about new development and new treatments for addiction is critical. So we are very grateful for your lecture, which was really fantastic. As a reminder for the audience, if you have a question, so again, please submit your question through the videocast window that says send live feedback, and I will repeat the question. And so I will start with the first question, Kim, the first question is from Dr. Roland Owens. And the question is, do the antibodies cross-react with endogenous opioids?
Which ones? Heroin, no. Could it endogenous opioids like morphine in the brain? Mm-hmm. Yeah, they could. But I think the level of these opioids is quite low, so I don't really think it would be an issue. I don't know if 6-acetyl morphine is made in the brain. I don't think it is. I think endogenous would be morphine. Some of the other ones, the peptide ones, no, they're not going to interact with those.
Okay, thank you. And another question is from Dr. Julia Johnston. And the question is, why would deuterium increase the efficacy of the vaccines?
That's a good question. In terms of the heroin one, the reason why I thought it would possibly do that is that change the overall rate of degradation of heroin. Deuterium is known to be more stable, and so I thought that would work with that. Why they could be better if you put them in proteins in that, as you can see, the immune system seems to recognize the difference between a deuterium and a hydrogen atom, and so it's recognizing it as foreign. So I think if you could incorporate these into protein-based vaccines, it could make them more immunogenic, being foreign, but would still recognize that bacteria or virus as being, what it is. So that's how I think they could improve upon a vaccine being a better vaccine.
Okay, thank you. And Kim, I did have a question myself. So in general, from a conceptual standpoint, when we think about an immuno-based approach for the treatment of addiction, do you think that this approach will be the most effective in preventing the development of the more severe forms, or it could be also effective in the patients who have already developed the more severe form of alcohol substance use disorder, like opioid use disorder?
Yeah, I don't know. You would have a better idea in that. It's hard to say. I don't know. I always tell people this is, I'm not selling you that this is going to cure everybody. And I don't think these vaccines are going to be useful for all drugs of abuse. I think you got to pick and choose your battles. And again, that's based on how the PK of the drug and how the drug interacts with the body. So I think, there's going to be chances where they'll work well and chances where they may not work as well. So I think I can't, that's a great question. I just really can't answer that.
Sure, sure. No, I understand. Another question from Chris Wanek. So, how about vaccines against the compulsive behavior in general, such as gambling, OCD, excessive hand washing?
Well, I guess you'd have to be able to pick out the particular, if you had a marker or protein that, you could say caused that you could develop something like that. I talked to George years ago about developing this for alcohol, and it's like, can't do that. But I looked at possible other potential agonists or antagonists that could be involved in addiction, so we could look at that. But I think that's rather complex. You could find other routes around these things. So I think it's probably not going to be of value for those things.
Mm-hmm.
Sure. I could probably use one for gambling, though.
Yeah. Okay. Another question from Dr. Perilla. So you presented some very exciting possibilities. Thank you for a fascinating presentation. Can you please comment further how clinically relevant pain control for vaccinated individuals, e.g., who then might need fentanyl in an appropriate surgical setting, might still work if you were to develop a vax structure that applied for various street formulations?
Yeah, the fentanyl ones we're developing wouldn't work in fentanyl, but some of the ones like alfentanil, remifentanil, things like this, we're not developing on that. So there are other fentanyl analogs that could be used in clinical ones that would avoid what we're trying to do. So definitely we had that in mind to try to, can't avoid everything, but to save a few on the side if you need to do it. You also have to remember with whether it's the vaccines, like it's the active vaccines or the immunization type ones, or if it's a monoclonal, the half-life of these things are only certain periods of time that they're going to wane off, and then unless you boost them back up or add it back in. So there aren't going to be windows of periods where they're going to fall off that you're going to be able to use some of the other drugs. So it's not like it's going to be set in stone for a lifetime. And the other thing too is if we're like with typical like a piece of bacteria or virus where it reinfects the immune system, you have B cells were alerted that are going to come back after it, or memory cells. It's typically not going to happen with these drugs. They're not going to typically, re-up the immune system. You're going to have to do that by boosting it up yourself. So it's not like these things are on there forever.
Sure, sure. One more question, Kim. So as we know, there are effective medications for addiction. We have FDA-approved medications for alcohol use disorder, for smoking, and for opioid use disorder. And specific to your work on opioids, we have a methadone, buprenorphine, and naltrexone. So the question is there any work from your lab or other labs to try to combine these approved medications for opioid use disorder with a vaccine-based approach, and see if the combination could be synergistic, for example?
Yeah, that's a great question, and it's something I thought about. But we could, in terms of the basic research, I think that's something we could do. But in terms of moving forward in the clinical trials, you're just doing the vaccine or the monoclonal, and to get that approved, then maybe you could pull the other one in. But in terms of trying to move both those things as a combo, I think that'd be very difficult to do. It's just like trying to develop a heroin and fentanyl vaccine. You're not going to be able, like I showed, combining those two and vaccinate it works. There's no way the FDA is going to allow that. You're going to have to get that fentanyl one approved and the heroin approved, and then maybe you mix them together. But you're not going to get them approved as a mixture. They're just not going to allow that. So I think that's a great question. I'm confident that you could use them in combination because the vaccines will have no effects on buprenorphine and naltrexone.
Right.
So I'm confident that combination would work, and it probably might work better, in terms of how these things would work. But in terms of moving forward, I think the first step would be just getting a vaccine approved, and then think about putting these other ones in. But people get all worried that this is going to replace these things. It's not going to. In fact, I would encourage probably putting it together. I think that would probably be like a great idea. I'm sure it would improve upon either one of them.
Sure. Okay, sounds good. It's 4:01, so we had to wrap up. I know people may have more questions. Feel free to reach Dr. Janda directly. I'd like to remind the audience again about the CME code for today is 37920. And in closing, on behalf of NIDA and AAA, on behalf of all the NIH community, Dr. Janda, thank you so much for your outstanding lecture today. And thanks everybody for joining today. And have a good rest of the afternoon. Thank you, everybody. Bye-bye.